Obtaining modified mononucleosome is crucial for designing epigenetic experiments. Our protocol provides acetylated, reconstituted mononucleosomes that can be used in biochemical and binding assays, structure determination, and more. The primary advantage of our technique is its versatility. This system can be easily modified to incorporate many types of modifications into mononucleosomes.
Begin by preparing 1 liter of autoclaved 2YT media in a culture flask. Inoculate 20 milliliters of the media containing appropriate antibiotics with the co-transformed cell stock that has the tag containing plasmid and PE-vol ACKRS, and grow them at 37 degrees Celsius to obtain an OD of 0.6. Add appropriate antibiotics in 1 liter of autoclaved 2YT media and inoculate it with 20 milliliters of starter culture. Grow the culture at 37 degrees Celsius for 2 to 3 hours to obtain an OD of 0.6 to 0.8. Add inducing agents and ACK for a final concentration of 1 millimolar IPTG, 0.2% arabinose, and 5 millimolar ACK. Grow the culture at 37 degrees Celsius for 6 to 8 hours. Then pellet the cells at 2700 times G for 15 minutes. And store them overnight at -80 degrees Celsius. Dissolve the pellet in 50 milliliters of histone lysis buffer per liter of culture. And sonicate the cells at 60% amplitude for 3 minutes with 1 second on and 1 second off intervals. Pellet inclusion bodies at 41,600 times.